Good morning, everybody. And it's a pleasure for me to be here today. I really applaud Ling and Bill for expressing their experiences here today, and also Ling for participating in the clinical trial. And we really encourage all patients and family members to think about clinical trials because that's where the future is. What I'm hoping to do is just walk you through some of the essentials of cancer development and some specifics about gastric cancer. But this is going to be a more pedestrian sort of presentation rather than show you a bunch of data that could be very boring for you. So let's... So these are data coming from the SEER database, which is from the United States. There are close to 80,000 patients. And you can see for all comers, all patients were diagnosed with stomach cancer. We don't really do well. So we lose almost 71% of those patients. And that is in contrast to Japan and Korea, where it's almost the opposite. And the reason for that is the early detection strategies that are in place, which are hard to implement in the United States because the incidence is quite low. So most patients for gastric cancer diagnosis are coming from China and Mongolia. So we have close to 1 million new diagnoses of gastric cancer, and 43% come from a specific region. Then, but if you look at per 100,000 cases, China is almost half that of Japan and Korea. So in other words, more patients per 100,000 persons are diagnosed in Korea and Japan, but more patients are diagnosed in China because of the high population. And I think the other problem is that majority of research is being done in the United States, in Japan, and some in Europe, but not so much in China. So they are beginning to recognize that to help their own population that is coming down with gastric cancer, they have to invest. And we are actually collaborating with 2 large institutions there. So I think things will change, and when China is engaged in researching gastric cancer, it will help patients everywhere, and I think the progress will become much more rapid. So one of the complaints we have is if you look at the caliber of gastric cancer on the society, the burden is very similar to a lot of other cancers, but they are common in the West. And so there is so much more research on colon cancer, prostate, breast, but not so much in gastric cancer. And I think this is why we are so happy that Department of Defense was able to allocate, basically entered the name of gastric cancer as an entity to be researched out of the $50 million pot they have every year. And that's because of Debbie's Dream Foundation and another entity that they were familiar with the Congress. So several people came together and we have that. Now we are after the NCI to allocate funding for gastric cancer. So now if we go to the NCI to get funding, we have to really compete with everybody else. There is no dedicated funding. And if you have an opportunity to address that, that will really be helpful. So the most established cause of gastric cancer is the bacteria. And I can't really point to you, but there is a bacteria at the top of that slide with the little lines coming out of it. And this is the H. pylori bacteria that likes to live in the stomach lining, but it hates the hydrochloric acid. The acid we make, this bacteria does not like it. So it buries down where there's no exposure to acid. So some of the processes through which this bacteria can cause cancer are shown here. Not everything is understood, but essentially it can make, certain species of bacteria can make a protein that it will insert into the host cell, in the gastric cell of the host, and it will change the composition of that cell to its own advantage. So it's not unique to H. pylori. All bacteria tend to do that. They manipulate the host for their own advantage. So one thing that this bacteria can do is stimulate the genes that allow the cell to multiply. Those are called oncogenes, and will also suppress the genes that can prevent proliferations. So you suppress the genes that can stop proliferation, and then stimulate the gene that allow proliferation. So that cells are starting to multiply. And this would be an advantage for bacteria, because then it will have many other cells to feed on, because it's constantly multiplying, and then you have many cells to feed on. But in the process of this, there are other things that are going on, which is they are damaging the DNA and causing chronic inflammation, production of other proteins and chemicals. And then I don't think the bacteria really wants the host to develop cancer, but the process continues to do that. So there are many moving parts to this. Not everybody with the bacteria is going to get cancer. There is your ability to repair the damage can prevent cancer. So that is something we have to understand. Okay, so there is some progress. There was close to a $300 million project from the NIH, from the federal government, from our tax money, which allowed us to investigate the very molecular genetics of gastric cancer. And we came up with 4 different types of gastric cancer. So now we can actually take tissue of the next new patient and classify the tissue into one of the 4 genotypes. And so we can take advantage of this information to some extent. There's much more work needs to be done. But if you look at the second column from your right, which says MSI high, if we identify a patient who has MSI high genotype, there is treatment. There's better treatment for them than we ever had before. And I'm going to explain that to you. So some work is being done, which is allowing us to understand in depth the composition of gastric cancer. So this is very fundamental. You think about the cell on the top, which is a normal cell, and you just think of that as the cell in the stomach, and then consequence of the bacterial infection could turn that cell into cancer, which is the cell in the bottom. So the cell on the top, the circle in the middle is called the nucleus, and all the red strings there, those are the genetic material. Half of that comes from our mom, half comes from our dad, and the cell is instructed to live by the code that is ingrained in the genome in the middle. So the genomic material is like simply a book. It itself doesn't do much, except you can read it. So if you wanted to change oil in your car, you would go to the manual, that will be the genome, but you have to change the oil yourself, that will be the proteins. So the cells work like that, and they're happy following the DNA instructions. But over time, which is the arrow going down to the cancer cell, the DNA can get damaged. So H. pylori can damage the DNA through chronic infection and reactive oxygen species and so on. So once there is critical damage, that cell is no longer normal. it becomes cancer. And cancer cells have the pressure to survive, and one of the survival tactics is to multiply. So then they will multiply. You can see at the bottom left, there are 2 daughter cells, and then one of the daughter cells is making more daughters and more daughters and more daughters. So the damaged DNA from the founding mother cell are being passed on to the daughter cells, and as the further generation, there's accumulation of even more DNA damage. So if you think about the granddaughter of the cancer cell, it is having much more DNA damage, and is much more aggressive, and is struggling to survive in the body of the host. But we shouldn't really get cancer, because we have evolved to have fantastic protective mechanisms. So one thing, we have a beautiful DNA damage machinery, because every cell in our body, which has 23,000 genes, the DNA is damaged every day 20,000 times, and it is repaired almost to the original state. So if there is H. pylori bacteria and causes DNA damage, we should be able to repair that. But obviously that doesn't happen. And if the DNA is not damaged, there is another machinery in the cell that will allow the cell to commit suicide. So the cell, if it is damaged and it's not repaired, then it should commit suicide. So we don't have an unhealthy cell in our body. Well, obviously that also doesn't happen. The third thing that should happen is the immune system should come around and find a sick cell and kill it. And obviously the immune system in a lot of our cancer patients is pretty healthy, and the cancer is growing very happily at the same time. So the immune system is very reluctant to kill cancer in a normal situation. Now, just to give you an example how good the DNA repair machinery is. So if you were to type at 60 words per minute, day and night, for actually 38 years, and you make one error in one of the words, one typographical error, that's how good our DNA repair system is. And yet, so many people get cancer. And this is done very fast because we have a very long DNA, 3.5 billion letters. So just to give you an example how quickly this repair is done and so efficiently is if you were driving the car at 387 miles an hour on a highway and still repairing at the rate that I told you in the previous slide. So very high speed, high accuracy. And I think this works better with the pointer, but let's try this. So first thing in the DNA duplication, which is required for proliferation, and the cancer cells have to have another set of DNA in order to have 2 daughter cells. So the first thing that happens is the DNA has to be pulled apart so that we can start to read it. The second thing would be to make a duplicate copy of the DNA. And finally, there is a proofreading. So although only occasional mistakes are made, we have a machinery, a protein that will come along and read the DNA, and if there are errors, then it will repair it. Okay, so now at the top, from your left to the right is the new DNA is being formed. So the pol E and the exo enzymes are basically copying DNA, and they're just going along making a new copy of the DNA. And then there are 4 proteins, MSH2, MSH6, and that group of proteins come behind the pol E and exo and will proofread and repair any mistakes. So you can see I think it's to your right that the good DNA is being made. Let's see. So this is the good DNA. And its DNA is perfect. It's a perfect copy, no mutations. But if you have, like in the middle, there is an kind of a lightning arrow on the proteins that repair the genome. If they are making a mistake because they have a mutation, and they can have a mutation, what is called the Lynch syndrome, where you get colon cancer, endometrial cancer, stomach cancer. If you inherit a mutation in those genes from your parents, then you're very likely not to be able to repair the DNA. Now you can see that on your right, there are a couple of yellow crosses. Those are mutations that this machinery has missed because it's inefficient. But at the bottom, if you have two mutations, one is in the DNA copying machine, and the second in DNA repairing machine, you end up with so many mutations. Now those people are going to get a lot of cancers because their DNA is very damaged. Okay, so I'm not going to go through this because it works very well with the pointer, but on the top you can see there is a mismatch pair. I think it's G and I can't read from here, but that two letters at the top, the middle of the lines, that's a mismatch in the DNA letters. And then the machinery comes down, and at the bottom, GC is the corrected thing. So if you have a mismatch, you can correct that, and that's called mismatch repair. And that relates to the MSI high, which you will hear many times now, because we need to know in almost every patient with advanced cancer whether there is defect in this machinery, because the drug called Pembro is very helpful there. So our DNA have these little satellites. A lot of this is, we don't understand why we have them. But the one from 1 to 9 base pair on your right bottom, those are called the microsatellites. And we have 19 million of these little sequences interspersed in our genome all over the place. And we don't quite understand what they do, except a lot of people think it has a lot to do with the immune system. But anyway, these are highly susceptible sequences for mutation, where the proteins that copy the DNA, they tend to slip off. So they just fall off, and they have to reload, and they fall off, and in the process, they can make a mistake. So that's why we focus on these microsatellites as indicator of the fault in the machinery. You look at them and say that this machinery must be not doing very well. Okay, so the way to understand is you have the DNA on this side, and you have the proteins that repair the DNA. You can look at the protein and say the protein is defective, therefore the DNA must be mutated because they can't repair. Or you can just look at the DNA and say there are a lot of errors, so this protein must be defective. So the MSI high testing is sometimes you can do the protein, sometimes you can look at the DNA. And it is a sort of a state, it's in flux because we need to figure out how to accurately figure out whether patient is going to benefit from immune therapy or not. Okay, so this is just one set of data where different tumor types are in the bars, and the level of the MSI high or mismatch repair can occur in the tumor. And the most common tumor that has a lot of repair problems is the uterus cancer. But immediately after that is colon cancer and stomach cancer. So those two bars after uterus are the tumor types that I deal with often. And this, I don't think you will be able to read, but in the previous set of data, colon cancer was the second most common. This is the data from another group, and here the stomach cancer is second most common cancer where there is defect in the repair of the DNA. So we are going to skip this. This doesn't work well without the pointer. So as I was telling you that you can do different types of tests to find out, and this is so important to know because the results are dramatic, and I'm going to show you those results. But on the top is you're looking at the proteins. In the middle, you're looking at these little microsatellites. And at the bottom, you're looking at the whole genome. You can sequence the entire genome and figure it out. Okay. So these are the data from patients who have tumors that do not repair the DNA properly. And what you see is the horizontal line in the middle is where the tumor volume was before treatment. And towards your right, all those bars going below that line, it showed the degree of shrinkage that occurred with immune therapy. So this is really dramatic. We don't have any other drug, like a chemotherapy drug, that can do this kind of reduction in size of the tumor. So this is a new development, a very promising one. And these are the survival data. Again, these are patients who have been heavily treated before. They have stage 4 disease, but they have mismatch repair defect, and they are treated with immunotherapy. And now you can see their survival is striking, and we have never seen the graphics like this before. Okay, so just a couple of minutes, I want to tell you how the immune system can recognize problems inside of the cell. So there's a lot going on inside of the cell, but the immune cells that survey our normal cells, they are actually touching the outside of, say, for example, cancer cell. So how do they know what's going on inside? And that is because if you have defective DNA, which I'm made with the little red lumps inside the DNA, those defective DNA will make defective protein. And defective protein, part of that is chopped up and is put up at the surface of the cell. That's what the immune cell will read. And it will say that this protein is defective, piece of that, and therefore something very wrong inside. So we have two types of these receptors. It's not really important to know today, but the type 1 is for the normal cells, and type 2 is for the immune cells. And I just want to point to the bottom of the slide. On your left, there is the red line, and that's what the immune system is going to read. That's basically a sequence of amino acids, and it will come and read that. That's part of the protein inside the cell that is being shown outside. And then when the immune cell will get suspicious, then something has to be done, and it will go to the killer T cell and tell the killer T cell that this sequence is not good for the host, and the killer T cell will have to make a decision. But normally, they are very reluctant to make a decision. Our immune system has evolved to be very reluctant when it's reading some of the sequences that are human. So the cancer has a lot of human sequences, obviously, because it derived from us. So the immune system is very inhibited. So here you have the very large cell, which is the surveying cell, is talking to the killer T cell. And this talk can go on for almost 18 hours before the T cell decides what it should do. So you can see there is a lot of hesitation there. Okay, so now we have 3 biomarkers that in gastric cancer patients we must determine. And I think I'm going to skip this part in the interest of time. So we can talk about the treatment options during the question and answer period. But we have increasing number of treatment options, and we're making some progress. I think it will come up here. So obviously some progress has been made because of the new frontier we have, which is to enlist the immune system to attack the cancer. And it is very capable of doing that. The only thing is it's highly reluctant to attack the cancer. And I think we obviously need to do more basic research, which will uncover the mechanism through which we can disable cancer from multiplying so rapidly. And obviously more effective treatments are needed because a lot of our patients are not doing well in the clinic. And they are very disappointed, and we constantly disappoint them and their families. Okay, I will stop here. Thank you.